In the first 9 months of our existence, something truly mind-boggling happens. From just a tiny cluster of cells, the most complex object in the entire known universe gets built, the human brain. Today, we're going to unpack the absolutely breathtaking story of how that happens. So let's just start with the big question, right? How do you build it? What's the blueprint? What's the construction schedule look like? And maybe most importantly, what happens when the whole process gets influenced by the world outside?
All right, here's our game plan. We're going to look at this as one big construction project. We'll walk through the incredibly precise timeline it has to follow and see what happens when that blueprint goes a little haywire. Then, we'll dive into the unseen influence of the prenatal environment, how it can literally rewire connections, and why our very first home matters so much.
Okay, so let's think of this whole thing as the ultimate construction project. It's this amazing sequence of overlapping phases, where each step perfectly sets up the next one. And it all has to start with laying the foundation. So the very first massive step is something called dorsal induction. And this happens when an embryo is just a few millimeters long. I mean, tiny. A flat sheet of cells, the neural plate, literally folds up on itself to create a closed tube. And this tube, this is the starting point for the entire central nervous system. We're talking the brain and the spinal cord. It is the absolute foundation that everything else gets built on. So, how do we get from that simple little tube to the complex, tangled network of a finished brain? Well, it happens on an incredibly precise and frankly unforgiving schedule. Every single phase has a critical window of time to get it right. So, right after that neural tube closes up, ventral induction kicks off. This is what cleaves the front of the brain into two halves, our future left and right hemispheres. Then comes an absolute manufacturing boom called neuronal proliferation, where billions upon billions of brain cells are created. Starting around week 12, those new cells begin this epic journey called neuronal migration, traveling to their final destinations. And finally, from 22 weeks on, the real detailed work begins, cortical organization. This is the intricate process of wiring the whole system together. But what happens if there's a glitch in the blueprint during one of these critical stages? Well, it turns out that the timing and the type of problem lead to very specific kinds of malformations. For instance, if that very first construction step, dorsal induction, doesn't complete, if that tube doesn't seal up all the way, you get what are called neural tube defects. It's literally like having a critical gap in the foundation of a house. One of the most devastating consequences is a condition called exencephaly. Now, diagnostically, as the caption notes, this can show up as what's called the Mickey Mouse sign. It's a strange name, but it's where the brain hemispheres actually develop outside of a skull that didn't form correctly. You can see on sonograms how the brain tissue is just sort of floating. It's a tragic but very clear illustration of what happens when that initial tube closure fails. Now, if the error happens a bit later, during ventral induction, the problem isn't about closing the tube, it's about dividing it. The brain might fail to split into two distinct hemispheres. This results in a condition called holoprosencephaly. And as you can see here, in the most severe form, known as allobar holoprosencephaly, the hemispheres are completely fused. I mean, just imagine, there's no dividing line at all, just a single, undivided forebrain. It's just a powerful example of how crucial that fundamental division process is. And yeah, errors can pop up at pretty much every stage. If not enough neurons get made during that proliferation phase, it can lead to microcephaly, which means a smaller head. If those neurons don't migrate to the right spots, it can result in lissencephaly, literally a smooth brain that's missing all its normal folds. And if that final wiring goes wrong, you can get something like polymicrogyria, a cortex with way too many small, disorganized folds. Okay, so it's crystal clear that having a precise genetic blueprint is absolutely essential. But it's not the whole story, not even close. The environment of the construction site itself, the prenatal environment, plays this profound and often totally unseen role. And that environment can be more challenging than we often realize. A huge recent review showed that a pretty significant number of pregnancies are affected by maternal distress. We're talking about 22% of pregnant women reporting increased symptoms of stress, anxiety, or depression. So this isn't some rare thing. It's a very common factor in the developmental environment. And this brings us to a really critical point. Our development isn't just about the genetic code we're handed. It's a dynamic, constant conversation between that code and the environment it's unfolding in. The conditions on the construction site can literally change how the blueprint gets read.
So, how in the world does something like maternal stress actually influence this construction project? Well, it's not magic, it's pure biochemistry. Stress hormones can cross the placental barrier, and when they do, they change the chemical environment for that developing brain. And what research is showing us now is that this doesn't just have some vague effect, it can subtly reshape very specific, very vulnerable parts of the brain. Study after study points to a whole network of regions that seem to be particularly sensitive. We're talking about the amygdala, our emotion processing hub, the hippocampus, which is crucial for memory and regulating stress, and the prefrontal cortex, the CEO of our brain, handling all the executive functions. Basically, the very circuits that are going to govern our emotions and our thinking for the rest of our lives. And the effects can be really nuanced. The thinking is that prolonged exposure to stress hormones like cortisol is a major driver here. For example, prenatal anxiety has been linked to changes in the actual size of the hippocampus and amygdala. Prenatal depression hits these same areas, but is also specifically linked to changes in the microstructure of the corpus callosum, that's the massive data bridge between the two hemispheres, and to weaker connections in key emotional regulation pathways.
So let's pull all this together and look at the big picture. What is all of this telling us? It's telling us that our development is a story written in two languages, the language of our genes and the language of our experience, which starts from day one in our very first environment.
And that right there is the key takeaway. The story of how our brain gets built isn't just a monologue written in the language of DNA. It's a dynamic conversation, a constant back and forth between our genetic inheritance and the environment of the womb.
Look, recognizing this deep connection isn't about assigning blame or creating anxiety. It's the opposite. It's about understanding and empowerment. It highlights just how unbelievably critical maternal well-being is, not just for her own health, but as a fundamental building block for the health of the next generation. Which, of course, leaves us with a final massive question. If the actual architecture of our brains and the very foundations of our future health are being laid down in these first 9 months, what more can we do, and what more should we do as a society to protect and nurture this incredibly foundational period of human life?